Hello, welcome back with a new background. Yesterday I was sitting there I was sitting, but now I have changed my setup to this side, facing the window on the opposite side. Anyways, yesterday we did complete 2 parts of water. Now we're going to be doing the rest. Disable. So hold on, let me just share the screen. So yeah. So yeah, let's just continue. We completed cohesion. Hold on, is my microphone open yet? So we completed cohesion and we completed adhesion. Now we're gonna move towards high specific heat capacity and high latent heat. So before just moving on, should I give a bit of a review, or maybe I should just give it at the end? Anyways, so what is a specific heat? This is a concept that is taught in physics, but I'm just gonna give you a brief overview of it. So specific heat capacity of a substance is amount of heat required to raise temperature of 1 gram of a substance by 1 degree Celsius. So what does it mean? So for example, here we have a glass of water at 16 degrees Celsius. Now, for example, the amount here we have is 10 grams of water. So we provide this system some heat, and because of that heat, the temperature of water goes from 16 to 17 degrees Celsius. Now, what is going to be the heat capacity of the, you can say, the heat that is transferred in water? So, 10 grams required. So, for example, we gave 500 joules of energy. So 500 joules over 10 is equals to 50 joules. So this is an example, yeah? So 50 joules energy is required for 1 gram of water to turn from 16 degrees Celsius to 17 degrees Celsius. This increase in 1 degree in 1 gram of water accounts for the high specific heat. Now, what's special about water is that it has a high specific heat capacity relative to other, you can say, compounds. For example, alcohol, which has lower high specific heat capacity. Now, there are two things that you need to understand here. Heat capacity and temperature are two different things. Or you can say thermal energy at this point. So thermal energy and temperature are two different things. So for example, this is a pool of water and it contains around 1,000 liters, whereas this is a glass of water and it contains 500 milliliters of water. So the thing to notice here is, even though this glass of water, okay, let me just mention the temperatures here. So this glass of water is at 50 degrees Celsius because we dropped it into the oven and or put it on the stove to heat it, whereas this is at room temperature. Yeah. So now the thing is, temperature is higher in the glass of water, but the thermal energy is higher in the pool. Now, why is that so? Thermal energy is a potential, yeah? So it's energy that accounts for the average kinetic energy of molecules. Total molecules, yeah? And because there are a lot of molecules in it, so the total energy, thermal energy, is going to be greater, but the temperature is going to be greater in the glass of water. So this was a necessary concept for you to understand. Now, why was that important here? So the thing is, this ability of water of having a high specific heat capacity, what does it do? It helps water to absorb high amounts of energy, of heat energy, without changing much temperature. Absorb or lose. So this property of water, of that it can absorb or lose high amounts of energy without much change in its temperature. This is very important for biochemical reactions occurring inside the body and also for the survival of the living things inside the ocean. For example, you can take an example of sea. Yeah. Now in the winters, or you can say in the summers, there is continuous sun rays on water, yeah? But due to its property of high specific heat, it absorbs a lot of energy without change in its temperature, without a significant change in its temperature, which is very important for the survival of marine life. Now, another concept that is linked to this is high heat of vaporization. Now, this accounts for the amount of heat required to change liquid state of water to vapor state. So, in other words, this is a glass of water again, and we boil it for half an hour, and primarily it had 200 ml of water, yeah, but due to significant boiling, we reduced its volume to, for example, let's say 100 ml. Now, the heat provided for the change in water, for change in its state, for the change of 100 ml water from its liquid state to gaseous state, this amount of heat is called specific heat of vaporization. And in the case of water, it is very high, which is 574 kilocalories per gram. Per kilogram, sorry. So why is it important? So for example, vaporization helps in as it provides a cooling effect on the surface that it's being evaporated on. So let me give you an example. For instance, this is a patch of skin here, and due to heat waves on it, it increases in temperature. For example, it increases from 38 to 39 degrees Celsius, which is higher than the body temperature, but this is an example here. So what happens is skin releases water molecules in the form of sweat here, yeah? And now these water molecules absorb the energy from the skin here. They absorb the thermal energy from the skin. Because of this absorption, the temperature of skin decreases from 38 to, for example, let's say 36. Now, what happens is the molecules that are already energetic escape due to the absorption of energy, and this release of atoms, or you can say molecules that are highly energetic, this decreases the temperature of the, you can say, the layer of water below. So what happens is, for example, if the complete water droplet evaporates, the temperature of skin drops because all the energy or all the thermal energy that was increasing the temperature of skin, that is absorbed by water, and the water has evaporated. So this is very important property of liquid here, as it's used in thermal regulation, and it provides a cooling effect when evaporates during transpiration and perspiration, or you can say in humans in the form of sweat, to regulate the body temperature. Now, so we're done with cohesion, adhesion, high specific heat, high heat of vaporization. Now we have ionization of water. So, what is ionization of water? So let's say this is a water molecule. It's a bit bented, but for better understanding, I've built it straight. So, a water molecule contains 2 hydrogen ions and 1 oxygen ion. No, not really ion, but atom, yeah? So, 2 hydrogen and 1 oxygen makes water here. Now, due to high electronegativity of oxygen, it pulls electrons towards itself, which weakens the bond of hydrogen with oxygen. Now sometimes it is rare, but it happens. What happens here is that the hydrogen bond breaks, and when it breaks, by donating an electron to oxygen, the product is OH negative and H positive ions. These ions, remember what is the contribution of these ions? OH negative ions are alkaline, as they accept electrons, protons, whereas this is the symbol of acidic nature, or acid. Now, this property of water to split into OH and H positive ions gives it a amphoteric nature, and it can also behave as a buffer. So because of this, it maintains the pH of enzymatic activities in cells and organs. So this is enough for, I think, this property, but I should write it for. And the pH of water is 7. You need to remember that too. Anyways, we're left with anomalous behavior of water. So now, a lot of elements or compounds show a property, which is that, for example, liquid A, this is a liquid A. When we cool it down, its molecules come closer to each other, forming a solid structure. When this compound is freezed at a specific temperature, so its compounds comes closer to each other, hence increasing the density. So remember, what is density? Density is mass per unit volume. So the volume decreases, and because of this, the density increases in other substances. But what happens in water here? Exactly the opposite. So in water, the temperature, when we decrease it, it behaves like other elements and other compounds, but at 4 degrees Celsius. Due to the hydrogen bonding, and I'm going to explain it, how does it happen, but for the overview, due to the hydrogen bonding, instead of volume decrease, the volume increases. And because of that, water is the only substance that if you freeze, increases volume. And below the 4 degrees Celsius, it behaves anomalous to other substances. So, for example, from 30 degrees Celsius, when the water is cooled from 30 degree to 4 degree, its density increases. But till 4 degree to 0 degrees Celsius, its density decreases. Now, this anomalous behavior is due to hydrogen bonding, and this has a very significance for life to exist. Now, why does it happen? So, for example, this is a water molecule, again, and this is a water molecule. Now, in liquid state, or I should just Now in liquid state, the hydrogen bonding in water molecules, between water molecules, is very random and irregular. And because of this irregularity, water molecules are at a specific distance here. But what happens when water molecules loses energy or are you can say taken to freezing point? So at below 4 degrees Celsius, instead of water molecules getting closer to each other, because of the property of hydrogen bonding, they form specific hydrogen bonding far away from each other at specific patterns. So they form hydrogen bonds at specific patterns with each other. And due to these patterns, these regular patterns, the volume of water increases. And due to this increase in the volume, the density decreases. And because the density decreases when water, you can say when water freezes, or you can say turns into ice, instead of sinking below, it accumulates on the top because of the low density. Remember the science concept that the thing that has low density than the liquid, or the solid that has lower density than the liquid, floats, whereas the solid that has higher density sinks below at the surface. So because of these regular patterns, volume increases, density decreases, and the ice floats on the water. Yeah. And because of this, for example, this is a swimming pool with a lot of fishes in it, and this is water here. Now the temperature here is, for example, 0 degrees Celsius, and you can say water starts to freeze. So this is formation of ice. So now if ice is formed, it starts going upside, or you can say the formation of ice starts from the top instead of from the bottom, because if water doesn't have this anomalous behavior and ice started accumulating at the bottom, then the freezing of oceans, or you can say freezing of this tank that contains fishes, should start from the bottom till the top when it's freezed. But what happens is, due to the low density, the ice accumulates on the top, acting as a insulating agent, because ice insulates ice acts as an insulating agent for this water body. And because of this accumulation on the top, not only the insulation is provided, but these fishes have water to swim in. So this is the concept here. So this anomalous behavior makes the life possible under frozen water. Now the last concept that was left is hydrophobic exclusion, and this should you can say clear out the concepts that were left in the lipids, in carbohydrates, in phospholipids, and it should be cleared now. So for example, this is a water molecule having a high electronegativity, and because of that the oxygen gains partial negative and hydrogen gains partial positive charges. Now this imbalance of, or you can say imbalanced share of electrons, and the electronegative atoms pulling electrons towards themselves makes this compound polar. Now, for example, there is another molecule which is polar here. For example, this is the head and these are the tails of, you can say, phospholipid that we just studied before. So when a water molecule is here, so it will form hydrogen bonds with the polar compound, because the polar compound has imbalance of electrons. So this hydrophilic head should be polar, having partial positive and partial negative charges on it. So these partial negative and those partial positive are going to attract each other, forming hydrogen bonds, and this is what leads to hydrophilic attraction. Now, what happens is, due to the you can say the hold on, let me just clear this out. So when these same water molecules comes closer to the hydrogen molecules, that these does not form you can say hydrogen bonds with the tails here. So because this molecule forms hydrogen bonds with the head and not with the tails are gonna be, you can say, repelled, because there is no imbalance of electrons, and electrons are present here. So for example, already present electrons, partial negative charge comes closer to the electrons here, electrons repulse each other, and due to no imbalance and everywhere where the density of electron being the same, the electrons repels each other, so the compound that is non-polar repels from the polar compounds. So this makes it insoluble too, because for example we have NaCl molecule here and water molecule here. So now what is going to happen is partial negative charge and partial positive charge, partial positive charge. So now partial negative charge is going to be attracted to the Na positive side, breaking the bond between Na positive and Cl negative, and that Cl negative is going to be attracted towards the partial positive side. So this will, what you say, dissolve NaCl in water. But if we take up non-polar compound, there is no attraction between water molecules and the compound, and sometimes there is repulsion due to the balance of electrons in non-polar compound, which leads to hydrophobic exclusion. So we're done with water. Now we're gonna start nucleic acids. But first, a small water break. God, I forgot to change the screen. What? Dude, I explained so much. Anyways, I am so sorry. I should complete it at the end too. I should revise it again because I didn't knew I forgot to change the tab. Anyways, so starting with nucleic acids. So nucleic acids were isolated by a Swiss physician, Friedrich Michel. From the nucleus of pus cells. And the thing about these compounds was that they were very different from other biological molecules, so they were named nuclein. And it was found that those same compounds that were named nuclein had acidic properties. And because of that, they were renamed to nucleic acids. Hold on, let me just check again. Yeah. So now, I feel terrible. I drew so many drawings to understand, and I literally forgot to change the tab to show this. Anyways, so this is what about is this is the history of nucleic acid. Now, the nucleic acids are polymers of 5 sugar-based compounds called nucleotides. So the monomer of nucleic acid is nucleotide, and basically all the nucleotides, like protein, have a similar structure, or you can say base structure, which is similar. So how does a nucleotide looks like? So this is a typical nucleotide molecule, and this is its first carbon, and this is its fifth carbon. So the nitrogenous base is attached at the first carbon, and the phosphate group is attached at the fifth carbon atom. So now there are two types of nucleotides, or you can say nucleic acids, based on the nucleotides. Number one is deoxyribose, and one is oxyribose. Or you can say ribonucleic acid. Hold on, let me just drink some water here. The heck? So now let's just continue. So you see on second carbon there is a hydroxyl group. So deoxyribose lacks this hydroxyl group, whereas ribonucleic acid, or you can say RNA, contains this hydroxyl group. So now there are four different, so now this structure, I'm gonna mark it. This structure is similar in every different kind of nucleotide. What's different, this is same in every different kind of nucleotide, but what's different is this. This is different. So what's the difference here? So there are different types of, you can say, rings, and it is not required in A-levels and any other board to remember the structure of these rings. But the only thing you need to remember is either if there is single ring or there is double ring. So the structures that contain double rings, like this one, are called purine, whereas the ones with the single structure is called pyrimidine. Now there are two different structures in purine and pyrimidine nitrogenous bases, and two different types in pyrimidine bases. So in purine it's adenosine or adenine, and guanine. whereas in pyrimidine it is cytosine and thiamine. Now let me just explain you a bit how they bond, how dinucleotides are formed. But you need to understand this. First, I should just tell you this, that if the complementary nitrogenous basis, there is another one which is called uracil, which is at the place of thiamine. Yeah, so in RNA, instead of uracil, instead of thiamine, it is uracil in the pyrimidine structure. So these adenine is complementary to cytosine, and guanine is complementary to uracil and thiamine. So what does it mean? So for example, there is adenine base. For example, this is an adenine base, and this is a thiamine base. They won't form any kind of bonds with each other, but if, for example, there is cytosine, sorry, I did it wrong, sorry. I did a bit wrong. Adenine bonds with hydrogen bonds with thiamine. What is happening to me? Anyways, A bonds with T, and if there is C, cytosine, so I should just remove it. Adenine bonds with thiamine and guanine bonds with cytosine. Now, what you need to understand is, if in front of A nitrogenous base, if C comes or cytosine comes, it will not form any kind of hydrogen bonds with C. But if T comes, it will form hydrogen bonds. Now you need to remember that AT forms 2 hydrogen bonds and GC forms 3 hydrogen bonds. Yeah, this is a thing to remember too. Anyways, how dinucleotide structure is formed. Now remember this phosphate group. This is very important in the formation of phosphate group. And we should just simply pick this structure here. So P double bond. So there are 2 consecutive nucleotides here. What? Should just build this. There is no difference, either you can put double bond O up or double bond O here. There's not going to be much difference, but for the convenience, for the understanding, and for not making much mess, I'm putting those double bond O here. So what happens is, these OH bonds, this hydroxyl group and this hydrogen, eliminates in the form of H2O, like we are used to watch in the proteins, in the carbohydrates, in the lipids, forming a phosphodiester bond. forming a dinucleotide. So this is a dinucleotide, and when there are a lot of similar dinucleotides with each other, this makes up a nucleic acid strand. You can say polynucleotide. So these polynucleotide chain is formed here, and it can vary in thousands of numbers. It can be of thousands of numbers, and for example, this is A, then this can be G, and remember, if it's C, then it would have only one single ring. So this is how polynucleotides are formed. Now, what about the structure of DNA? So the structure of DNA was explained by Watson, James Watson, and Francis Crick. And they proposed that DNA is double helix structure. Each helix is made up of 4 types of nucleotides. Both helix are complementary to each other. If one helix contains A, the other opposite or complementary helix would contain thymine, and if it's cytosine, the complementary would contain guanine. Each helix is composed of 2 parts: upright made up of deoxyribose sugar and phosphate, rung made up of nitrogenous base. Both helix are held together by hydrogen bonds due to the zipping and unzipping of both helixes. By making and breaking, there are hydrogen bonds. Both helix are opposite in direction. One chain runs from 5 to 3rd, whereas the other chain runs from 3rd to 5 in the other direction. The distance between the helix remains same from one another, i.e. 20 Armstrong. Each turn of duplex contains 10 base pair. So what does it mean? Let me just explain this to you. Delete. And listen this carefully, because an important concept is coming here. So this is one strand. This is another strand here. This is another strand coming here. So for example, this is a kind of DNA which is formed from 1, 2, 3, 4, 5, 6 nucleotides. And the nitrogenous base is 1, A, C, G, C, and A, T, for example. So these are the types of nitrogenous bases here. Now, what happens is it shouldn't be really like that, but for example, So you get the point. This is dash, dash, and dash, similar structure formed here too. So now and blah blah blah. So in order for the formation of hydrogen bonds or the double strand formation of the DNA, what needs to happen here is the nucleotides, which are complementary to each other, C, G, T, and A. So the nitrogen bases that are complementary to each other, that bonds with each other, needs to be complementary, needs to be opposite to each other in order for that chain to form bonds. Now, one thing that you should one thing you can see here is that T is opposite here, ATG, C, A, and ACG, C, AT. So now these both are complementary to each other here, and they form bonds. So A formed two bonds with two hydrogen bonds with thymine. C forms three hydrogen bonds, two hydrogen bonds, three hydrogen bonds, two hydrogen bonds, and two hydrogen bonds here. So this formation of two strands makes DNA look like this. So this is a visual representation of strands, and another complementary strand comes here which is opposite. For example, this one runs from third to 5, so this is gonna run from 5 to third. This is complementary, so they're gonna run opposite here, and they're gonna form bonds, and after that this similar would look like something like this, and this formation will then form kinks and twists, which will make what DNA looks like, a helical form from these double strands. So this is the typical structure of DNA. The formation of DNA, the formation of polynucleotides, dinucleotides. Now there are certain concepts regarding the DNA which we will discuss in the chapter of the genetics, because the structure of messenger RNA, the transfer RNA, the ribosomal RNA, these all are included in the chapter of genetics. Yeah, and the processes like translation and transcription, these are also present in the genetics. But for this chapter, what you need to know is the basics, the concept of DNA formation, the bonding between DNA, the phosphodiester bonds, the formation of the backbone, which is typically this phosphodiester chain is called the backbone of DNA. And this is very important. This hydrogen bonds refers to the if these are broken, this is called unzipping, and if they form, this is zipping of DNA. And DNA exists in these two forms. And now if it's RNA, these OH groups on second carbon are not going to be present here, yeah? So this is what makes RNA, and I think there is nothing much left in this chapter to discuss. So this was nucleic acids, and now the only thing left is some conjugate molecules that we need to discuss here. So from the name you can guess glycolipids, these are conjugate. So what is conjugate molecules? I should explain you first. Conjugate molecules are formed when biomolecules of two different groups combines chemically with each other, acting as a one unit and have a specific function here. So there are different types, but let me just give you glycolipids, glycoproteins, and lipoproteins, and nucleoproteins. So this is not included in A levels, but it is required in the medical tests, aptitude tests. So you need to learn it. So glycolipids are also called cerebrosides. So let me just tell you verbally what these are used for. What do they hold as their function? So these cerebrosides are conjugate of lipids. From the name glycolipid, they are formed from lipids and carbohydrates. They are also called cerebrosides because they are present in the white matter of the brain and myelin sheath of nerve fiber. So they are present in white matter of brain and they are present in myelin sheath of nerve fibers. So they have important function in the nervous system, nervous coordination. They're also present in inner membrane of chloroplast. So second, glycoproteins or mucoids. Glycoproteins are also called mucoids, and they are formed from combining the molecules of carbohydrates and protein molecules. Most of the oligo and polysaccharides in animals and plants are linked covalently to protein molecules, and there is an important function regarding that because they act as transport proteins, receptors, antigens of blood groups, etc. Because I mean they are part of egg albumin too, and I think they're present in gonadotropins too. In the chapter of cell membranes, we're gonna learn in deep how and why they are important glycoproteins for cell recognition, for antigen, etc. Anyways, coming to lipoprotein, they are a conjugate of lipids and protein as per their name. They help in transportation of lipids in blood plasma, and they occur as a component in the membrane of mitochondria and the plasmic reticulum and nucleus, egg yolks, and chloroplast membrane. Therefore, and the last one is nucleoprotein, which is formed from basic protein and nucleic acid, as from the name you can guess it, and they are main components of chromatin material, chromosomes, and ribosomes. So we're gonna learn about this, but let me just give you a brief overview on that nucleoprotein. That nucleoprotein, for example, in DNA is not present in the straight chain form. Yeah, it occurs in coil forms, and after these coils are then coiled over a protein molecule. So these double strands are coiled, and these coils are coiled in the protein, and this forms a nucleoprotein. Yeah, so the chromatin material that contain histone protein and nucleic, and you can say nucleic acids are the part of DNA. This is what makes up the nucleoprotein here, and they're also present. And you can say nucleoprotein is also the part of ribosome because ribosome is made from protein and RNA. So this is the ending of this chapter. We have completed successfully in, I think, how many hours? Let me just see. Oh, hold on. I should just explain the remaining part of water, which I just skipped a bit. The ones who understood it can leave. So I think specific heat capacity wasn't needed to explain much, but here we go. So in liquid water, the water molecules are present in irregular shapes. Yeah, they're present in irregular patterns. They form hydrogen bonds in irregular patterns. So their volume is, this is their volume. Due to irregular, it is less, but in the solid form, they form hydrogen bonds in patterns. You can see this. They are much more in volume. They're taking much more place than these. They are more compact. And because of this increase in volume, the density decreases and blah 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 what I said. I think there's not much need to explain now because I'm tired too. So anyways, you can see the notes that I drew here. And I think I would just explain this 2 to 3 topics of water, the high specific heat and heat capacity, if some of you didn't get it. But we will also start the chapter of cells next week, because this week we had done with biological molecules, and until then, bye bye.